All right, let's start. Okay. So in the last class, we were discussing about the photosynthesis, and after this class, we'll continue the biochemistry as well. Okay. From today, please don't keep any absentees. Please attend every single day without fail. Okay. Slowly, I'll increase the number of hours, 4, 5. By that, we have to work hard these days, especially. All right, let's continue. So in the last class, we were discussing about the cyclic photophosphorylation as well as the non-cyclic photophosphorylation, correct na? Which is the last electron donor, acceptor that is involved in the production of NADPH? Which is NADP plus, correct na? Which is the first electron acceptor that accept the electron from the P700? Which is the A0. Anyone remember A0 is also called as? Very good. A0 is also called as modified chlorophyll A. And what about the A1? Remember, A1 is also said to be the phylloquinone. Right, perfect. Okay. And what is the name of the amino acid that act as a final electron donor for the P680? What is that? Tyrosine. What is the side chain of the tyrosine? Good. What is that? Which is containing generally OH group. What is the major ions that are required for the photolysis of water apart from manganese? So we require the chlorine also, as well as there is a requirement of calcium as well, correct na? And what is the major force that is involved in the production of ATP? Which is proton motive force. So here I want to highlight some of the important points. While solving the PYQ also, I will explain. Okay, please continue everyone. Okay, now you remember how does this particular ATP is getting synthesized. First P680 accept the electron, eject the electron, and who will accept first? Pyophytin. And from the pyophytin, electron sent to plastoquinone. Now please remember all of you very correctly. There are two type of quinones as we read. One is called plastoquinone A and plastoquinone B. I want all of you to remember this important statement. Plastoquinone B can act like a mobile electron carrier, whereas plastoquinone A cannot act as a mobile electron carrier. It is acting as what? Immobile electron carrier. I don't know why my voice is fine, everyone. Okay, now remember that while solving the PYQ, we'll get a clear-cut idea. Right. Okay, so that is what light reactions and little concept is left out. Once we complete the dark reaction, we will come back. Okay, so all of you focus, online people also. Okay, now what is the ultimate goal of the light reactions? Ultimate goal of the light reactions, production of energy, which is ATP and NADPH, correct na? NADPH also act like reducing equivalent and electron carrier. This is most important, always remember. Whether it is a NADH or whether it is a FADH2, always they are acting like a electron carriers. That is carrying the electron or transferring the electron from one electron carrier to the other. Now, do not confuse in the exam if they ask you a question, photosynthesis is, what we can choose the answer? See, remember, they may give the option of oxidation and reduction, or they use the word redox reaction. Is it true or false? So we can say it is a redox reaction or oxidation or reduction. Now the question is very clear. Now if I ask you a question, production of glucose is, which is reduction. Now photolysis of water is taking place where? Is it in the reduction or oxidation? So photolysis will happen in the oxidation. We generate the energy. By using that energy, we are producing the glucose. But in simple words, production of glucose is always reduction. Photosynthesis is oxidation and reduction. Sometimes in the exam, instead of using the word oxidoreduction, photosynthesis is just a dash reaction. They can say redox reaction. Okay, always remember that. Now anyway, we finally produced abundant amount of the energy that is required for the plant to produce the sugar. Correct na? So now from the light reaction, plant shifted to the dark reaction. we already discussed that dark reaction doesn't mean that we require the light. It is what reaction? Light independent reaction. Whether the light is present or absent, it doesn't matter, right? Now we also call it as carbon fixation, because ultimate goal of the plant body is fixing the carbon into glucose. How they can fix the carbon into glucose? By assimilating the carbon. In other words, by incorporating the carbon into the plant body, by that it is reducing into the glucose. That's why we call it as carbon assimilation or carbon fixation or light independent reaction. Now all of you answer, where these dark reactions are taking place? Location. Why only in the stroma? Why not on the thylakoid membrane? See, I believe every one of you observed this picture in the last class. Why? Okay. Okay. Now what you people can observe here, NADPH as well as ATP. They are directly releasing into where? Stroma. Not only that, the enzymes required for the dark reaction all are present only in the stroma. So the question asked in the set exam, very simple question, what are the major enzymes that are present in the stroma which are required for the dark reactions? What are those enzymes? Number one is Rubisco, correct na? And Rubisco is present in which cells? We'll discuss that. But you tell me, Rubisco is present in mesophyll also, bundle sheath also, but varies from a plant to the plant. And what else we are having? Another key enzyme, PEP carboxylase enzyme. It is also majorly present in the mesophyll cells only. Okay, remember the enzymes compulsory. All right, look into this. Whatever the ATP and NADPH that we produced in the light reactions, entire energy can be used by the carbon dioxide, and the carbon dioxide is getting reduced into the sugar. Now you can observe the picture so clearly. The carbon dioxide cannot reduce into the glucose directly. First, it is forming what phosphates? Triose phosphates. Now remember always, first carbon give rise to 3 carbon containing compounds. 3 carbon containing compounds finally converted into the 6 carbon. So in order to reduce the carbon into glucose, we require only energy. We do not require the light. That's the reason we call light independent or dark reaction. All the plants present on this planet Earth broadly categorized into 3 types based on the dark reaction or based on the carbon fixation reaction. They are categorized into C3, C4 and CAM. So which plants are more predominant? Definitely C3. What we can say that 95 to 97.5% of the plants present on this earth belongs to C3 pathway. And do not confuse between the C4 and CAM pathway. Always remember in this way, the plants which are living at the high light intensities. In other words, we can remember all the plants which are generally monocots. In more clear way, more than 18,000 of angiosperms follows C4 pathway. So you can remember the plants which are living in the high light intensity or high temperature. I am not using the word desert temperature. I am just using the word high light intensity and high temperature. Whereas you all already know that the CAM plants nothing but what? Belongs to a family called as Crassulaceae. Nothing but desert plants. So there are 3 different type of plants, C3, C4 and CAM. Okay, everyone written 2025 June exam, no? The question came from which cycle? The question came from C2 cycle. Correct, na? So which is a part of C3 cycle only. We gonna read this. Okay, so now all of you understand the point very correctly here. What's the ultimate goal? Fixing the atmospheric carbon into sugar by using energy, which are generated during the light reaction. All the reactions are exclusively taking place in the stroma, okay, because of the availability of enzymes. Now we'll discuss slowly, because as I said, this is completely conceptual. You no need to read and remember a lot of information here, just understand. Our ultimate goal is fixing the carbon into sugar. This process or the Calvin cycle or the C3 pathway happens in 3 simple steps: fixation, reduction, regeneration. We already used a word, we can't fix the carbon into glucose directly. What we have to do it? First we have to fix the carbon into 3 carbon containing intermediate compound. That's why we call it as fixing. Then the same 3 carbon we reduce and same thing we regenerate. Let's understand step by step, slowly and elaboratively. Okay. What's the answer for this? Quickly answer. Don't take too much of time. Whomever didn't attend yesterday class, watch the record. Otherwise, you won't understand this class. Quickly. No, it won't be available in the Telegram. Still many people are. Let's see. Where is Travani? She didn't come. See, the question says that online people, electron from P680 moves first to what? Pheophytin, then blastoquinone followed by cytochrome B6F and plastocyan. So wrong option. P700 can receive electrons from, so plastocyan is the final electron donor. NADPH is produced at the end of light reaction. The hydrogen ions create a force called proton motive force. The all protons are coming from stroma to the lumen. So they finally accumulated in the lumen. Again they go back to the stroma. So that is also right. Hence the correct answer will be the option D. Okay. We have to go little faster. Okay. I'll explain this. Focus all of you correctly. As we all discussed very clearly that our ultimate goal of dark reaction is fixing the carbon into glucose. This is our ultimate goal. Now remember, in order to fix the carbon into glucose, how many times the cycle has to turn? We know very well glucose is how many carbon? 6 carbon. So definitely cycle also has to turn 6 times. So in order to fix one glucose, cycle turns 6 times. In order to fix half glucose, cycle turn 3 times. So what is the best way to remember? First you have to remember for single cycle. Then we will apply it for 3 cycles. Then we will apply for 6 cycles. Then it is easy to understand. Okay. So now look into the picture. We have to fix this atmospheric carbon into glucose, which means someone has to accept this carbon and convert it into the glucose. So who is the carbon acceptor in the C3 pathway? The name of the carbon acceptor is what? RUBP, ribulose 1,5-bisphosphate. How many carbon containing compound? It is 1, 2, 3, 4, 5. Correct na? So why we call bisphosphate? There are 2 phosphate groups are there at the first carbon and 5th carbon. So RUBP, which is a 5 carbon containing compound, from the atmosphere it accepted 1 carbon. So 5 plus 1, 6 carbon. But unfortunately the 6 carbon containing compound is highly unstable. Since it is unstable, it has to break down into simpler forms. And according to the rule, it has to break down into a simpler form, which should be always 3 carbon. Okay. So how many carbons we have? 6 carbon. I am having 6 rupees in my hand. I want to purchase the chocolate of 3 rupees. How many chocolates I can purchase? Definitely I can purchase only 2. So I have 6 carbon. I want to produce an intermediate which should be a 3 carbon containing compound. Correct na? Then I will get 2 molecules of 3 PCA. Now there is no confusion at all. So for 1 cycle, how many 3 PCA we will produce? 2. For 3 cycles, 6. For 6 cycles, 12. Correct na? So now first we will understand it for the 3 times and we can multiply for the 1 molecule of glucose. So for 1 round we require 1 RUBP. So let's consider initially for half glucose. For half glucose, cycle has to turn 3 times. So 1 round we get 1 RUBP and 1 carbon. So 3 rounds, how many RUBP we will get? 3. So 3 into 5, 15 carbon, because every RUBP is 5 carbon. Every RUBP accept 1 carbon. So 15 plus 3, 15 plus 3, 16. 15 plus 3 how much? 18 carbon. So, but what is our rule? Our stable product or intermediate product should be how many carbon? 3 carbon. So, I am having 18 rupees now. I want to purchase the chocolate of 3 rupees. Then how many chocolates I will get? We will get only 6. Correct na? This is what we call carbon fixation. Means we are fixing the carbon into an intermediate. That intermediate is how many carbon? 3 carbon. That's why name of the pathway is C3 pathway. And what is that particular intermediate compound? It is called as 3-PGA. Phosphoglycerate or phosphoglyceraldehyde or phosphoglycerate you can consider. Now don't confuse, each PGA is how many carbon? 3 carbon with 1 phosphate. Now this is a simple fixation step. Who catalyzed this step? The versatile enzyme Rubisco. Now the 6 molecules of 3-PGA in the next step, what is the next step? Reduction. Now understand all of you, the 6 molecules of 3-PGA is getting reduced to 6 molecules of G3P. What's the difference between PGA and G3P? Suppose consider this is the only question in the upcoming exam. What is the difference? Tell me. The difference between 3-PGA and G3P. Definitely there is one phosphate group on the 3rd carbon. There is no change. Look at this. 3-PGA contain what group? Carboxyl group. G3P contain aldehyde. That's all. So one 3 carbon containing compound reduced to another 3 carbon containing compound. So how many 3-PGA we have? 6. 6 molecule of 3-PGA got reduced to 6 molecules of G3P. There is no change in the carbon number. But in order to undergo the process of the reduction, we require abundant amount of energy. So if I want to reduce 1 molecule of 3-PGA into 1 molecule of G3P, I require 1 ATP and 1 NADPH. Since I am having 6 molecules, so 6 molecule of 3-PGA reduced to 6 molecules of G3P. Correct na? Then I have to use 6 ATP and 6 NADH. This step is called reduction. Means we just reduced from one form to the another form without changing any carbon number. Now slowly think and understand it correctly. Now after the reduction, how many G3Ps we are left over after reduction? We have 6 molecules of G3. Each one is how many carbon? 3 carbon. Out of 6 molecules of G3P, only 1 molecule of G3P exit the cycle. Only 1 molecule and produce half glucose. Tell me why half glucose? Because G3P is how many carbon? 3 carbon. In order to produce the glucose, we require 6 carbon. So we have only 3 carbons availability, hence we can produce only half molecule of glucose. Now tell me how many G3P are left over? 5. 5 means how many carbons are left over? 15. 5 into 3, 15 carbon. So a simple question, by using 5 molecule of G3P or 15 carbon, how many RUBP we can regenerate? 5. Why? Each RUBP is how many carbon? 5 carbon. But how many we are having? Only 15 carbon. We cannot generate 5. How many we can generate? Only 3 molecules of RUBP. Because each RUBP is a 5 carbon containing compound. How many we are regenerating? 3. While we are regenerating the RUBP, every cycle we use 1 ATP. Since we regenerated 3 RUBP, we can produce 3 molecules of ATP. So let me quickly summarize. In order to produce the half glucose, cycle has to turn 3 times. And every time the carbon can be accepted by the RUBP. So one time 1 RUBP accept 1 carbon. Since the cycle is turning 3 times, 3 RUBP accept 3 carbon. 3 RUBP is 15 carbon, accept 3 carbon from the atmosphere, total of 18 carbon, which is completely unstable. Hence it is breaking down into 6 molecules of 3-PGA. Now the 6 molecule of 3-PCA, each one is a 3 carbon with 1 phosphate undergoing the process of reduction and giving rise to the 6 molecules of G3P. For one 3-PCA wants to reduce into one G3P molecule, it requires 1 ATP and 1 NADPH. Hence we use 6 ATP and 6 NADPH. Now out of 6 molecule of G3P, only 1 molecule of G3P exit the cycle and produce half glucose. The leftover 5 molecule of G3P, which is total of 15 carbon, are going to regenerate 3 molecules of RUB by spending 3 ATP. Correct na? Now everyone understand it clearly? Now simply answer the question. Everyone calculate and tell me. Consider this is only the question. How many number of carbon dioxide, how many number of NADPH, how many number of ATP are required to produce 4 molecules of glucose and 2 molecules of sucrose? All of you answer? Everyone, online people, please answer the question. Once again, look into the given question. I am asking total how many number of carbon dioxide, NADPH and ATP are required for the 4 molecule of glucose and 2 molecules of sucrose. Try everyone. Tell me Bindu, what is the answer? Tell me Lata, what is the answer? Everyone online? What's the best way? First calculate for one molecule, then automatically you can get for all. Try it. Slowly, slowly understand. How much? 24? Okay. 48, NADPH? This is for what? 4 molecules of glucose. And for what about 2 molecules of sucrose? So which is same. Okay. So now how to calculate? First calculate for 1/2 glucose, then multiply for 1. For a half glucose, cycle has to turn how many times? 3 times. So we require 3 carbon dioxide. For half glucose, how many ATP we require? 9. We do not require 6. Here 6 in the reduction, 3 in the regeneration. So we require 9 ATP and how many NADH? 6 NADH. So then you can calculate for 1 glucose, which is 6 carbon dioxide, 9 ATP, I'm sorry, how much? 18 ATP and 12 NADH. So multiply into 4, which is 48, and multiply into 4, 72, multiply into 4, which is 24. Correct na? So you always remember for one molecule, then it is easy to answer for all other. Okay. And I also asked you for 2 molecules of sucrose. Actually, one sucrose is made up of what? One sucrose is made up of 1 glucose plus fructose. Anyway, later fructose is getting converted into the glucose only. Hence, we can consider 1 sucrose is generally made up of 2 glucoses. And I asked you for 2 sucroses, which is definitely 4 glucose. So, which will be the same. Correct, na? Right. Okay. So, now slowly understand. So, think properly and give me a valid explanation why this is your answer. Justify your answer. Answer this. Tell me correctly why. Vanilla, what's the answer? Why? Others? Online? Quickly, forgot everyone. Okay, what is the question? What is the net synthesis of on carboxylation of 3 molecules of RUBP in the Calvin cycle? What is the total number of, what exactly the question is saying? First give me the answers, guesses. Lakshman, what is the answer? How much? Niranjana, what is the answer? And others? See, what is the question? What is the net synthesis of G3? The word net synthesis representing how many number of G3P are directly involved in the production of glucose. So, if you observe the cycle, how many are directly involved in the production of glucose? Only one molecule, correct na? So, answer will be 1. See, what is the net synthesis of G3P and carboxylation of 3 molecules of RUBP in the Calvin cycle? Total how many we get? We get 6. But out of 6 only 1 exit the cycle and produce the glucose. Hence the net will be always 1. So always remember this thing very clearly. 9 ATP are converted to 9 ADP during the reduction and regeneration. 6 NADPH are used in the reduction. So you should always remember this. To make 1 molecule of glucose, we require 6 carbon dioxide, 18 ATP and 12 NADPH. So which you have to multiply accordingly. Okay. Now quickly answer. Anyway, we don't consume any kind of energy directly or indirectly in the fixation. Correct na? We spend energy, that is ATP and NADH, only in the reduction. Only in the reduction only we use NADH. But in the reduction also and even in the regeneration also we will consume the ATP. Okay, don't confuse. Okay. Now what we discussed, out of 6 molecules of G3P, 1 molecule of G3P, which is how many carbon? Which is a triose phosphate, exit the cycle and produce glucose. Now we all know, and we're going to read it in the transport in plants, none of the plants never ever transport glucose via phloem. Correct na? Plant always transport what? Sucrose. Correct na? So definitely the glucose whatever synthesized during the photosynthesis, it has to be converted into sucrose as well as starch. Sucrose is what type of a saccharide? Disaccharide. Starch is polysaccharide. Again the question for the exam, the locations. Always remember, sucrose is exclusively synthesized in the cytosol. Cytosol is the location. We will see in the upcoming slide. There is an important enzyme called sucrose synthase enzyme. That's the reason sucrose only synthesized in the cytosol. Similarly, in the chloroplast stroma, we are having a starch synthase enzyme. That's why it synthesizes over there, right? This is simple C3 cycle. Now, the most important in your 2025 paper also asked question is C2 cycle. So, before understanding the C2 cycle, what is this photorespiration and why it will happen? For that, we have to understand about an important enzyme which is called as Rubisco. Okay. What is the full form of the Rubisco? Ribose 1,5-bisphosphate. So definitely it contains 2 phosphate groups on the first carbon and 5th carbon, carboxylase and oxygenase, which means it is having the ability to fix the carbon also, ability to fix the oxygen also. But originally plant always required to fix only carbon. There is no point of fixing the oxygen because plant anyway releasing the oxygen. Hence it has to fix only carbon. If it fixes carbon, then only plant can produce glucose. If it is not fixing the carbon, if it is fixing the oxygen, glucose production will stop. It is definitely a harmful reaction or dangerous reaction for the plant. Now firstly, we have to understand why this Rubisco is fixing the oxygen instead of the carbon. Why does it happen? What is the exact reason? Now look at this. Rubisco fixes only oxygen at high light intensities and high temperature. Now think about it, why? Because when the temperature is higher, what happens? See, now this is the enzyme Rubisco. What is this site? Active site. In the exam multiple times asked a question, the enzyme Rubisco having multiple catalytic or active sites? No, it is having only one catalytic or active site to which carbon dioxide and oxygen is competing. So whichever is more available in the quantity, that will go and combine with active site, then the Rubisco can fix it. You're getting? Rubisco cannot differentiate which is the carbon dioxide and which is the oxygen. I mean to say in terms of active site. Whichever it fixes, it will catalyze that specific reaction only. But why only oxygen will go and combine with the active site instead of the carbon dioxide? The reason is at a high temperature. What happens at the when the temperature is high? When the temperature is high, carbon dioxide is available and oxygen is also available. But carbon dioxide is completely solubilized. Means there is no availability of the carbon dioxide at high temperature. Then only available is what? Oxygen. Okay. See, which is completely opposite in case of our human body. We read in the human physiology, out of 100% of the oxygen, you remember, only 1.5% of oxygen will be combined with plasma. Remaining 98.5 will go and combine with hemoglobin. You are understanding? But what about the carbon dioxide? You remember? Out of 100% of the carbon dioxide, at least 6 to 9% of the carbon dioxide dissolved in the plasma. Some percent combined with hemoglobin. But here what is happening, whatever the carbon dioxide is available, that is getting completely solubilized, which means no carbon dioxide is available. What is available? Oxygen also solubilized, but a little quantity is left out. So Rubisco uses that oxygen. When Rubisco fixes oxygen instead of the carbon dioxide, that reaction is called as photorespiration. It is not at all necessary for this reaction to be happened. Correct na? Because plant has to fix only carbon, but unnecessarily it is fixing the oxygen because of the activity of the enzyme at a higher temperature. Now look into this important line. Oxygen and carbon dioxide compete for same. So there is only one active site. There are no two active sites separately for carbon dioxide and oxygen on the Rubisco. Now tell me, you only answer this. So who is the carbon acceptor? RUBP. RUBP accepted carbon now. What is the enzyme now? Rubisco. So to the active side, who will go and attach us now? Carbon dioxide, because availability of the carbon dioxide is more. Then how many intermediates we get? How many intermediates we get? RUBP is how many carbon? There is one carbon dioxide from the atmosphere. Total 6. How many intermediates we get? 2. When I use the word intermediate, always remember it is 3 carbon. So how many intermediates we get? 2 molecules of 3PGA. Can I say Calvin cycle continues without interruption? Correct na? Because we fixed only the carbon. Now the problem is that because of the high temperature, the carbon dioxide availability is extremely lower. Then RUBP combines with what? Oxygen. Now Rubisco can fix only what? Oxygen. Now how many carbons we have? 5 carbon. How many intermediates we get? Why? Why we get only 1? Because I have 5 rupees in my hand. I want to purchase a chocolate which is of 3 rupees. So how many chocolates I can get? Only 1. So I have 5 carbon in my hand. So how many intermediates I will get? Only 1 intermediate I will get. 1 molecule of 3PGA. How many carbons are left out? 2 carbons are left out. That molecule is called as 2PGA. 2-phosphoglycolate. Is it necessary to produce this 2-phosphoglycolate? It is not at all necessary. Our ultimate goal is what? To produce 2 molecules of 3PGA. But because of the activity of the Rubisco at the high temperature, we produced only 1 molecule of 3PGA and 1 molecule of 2PGA is how many carbon? 2PGA is how many carbon? 2 carbon. That's why photorespiration is also called as C2 cycle. Okay, since the end product is of a 2 carbon containing compound. Now tell me, when the RUBP fixed when the Rubisco fixed the oxygen, Calvin cycle will continue or not? No, it cannot continue. Hence the plant cannot produce the glucose. Then what plant has to do now? It has to convert this 2 PGA back to 3 PGA. Then only the cycle will be continued. Correct na? So how this will happen? How plant will convert 2 PGA to the 3 PGA? Okay. So this is the question asked multiple times in your very recent question paper also. So don't confuse, once again understand, this is RUBP, which is a 5 carbon containing compound. Combined with what? Oxygen. How many 3 PGA I will get? How many 3 PGA I will get? Only one. How can I get 2? Because it is combined with what? Oxygen. In the presence of Rubisco, I will get 1 3 PGA. And what else I will get? 1 molecule of 2 PGA. That's all. Now what should I do now? I have to bring this 2 PGA back to 1 molecule of 3 PGA. Then only the Calvin cycle will be continuous. Correct na? For this, there is an involvement of multiple organelles. We generally called CPM. Chloroplast, peroxisome and mitochondria. And we cannot bring back this 2 PGA into 3 PGA in one single step. It takes multiple steps. This is the exact question asked in the exam. Now all of you focus, what's happening now? First 2 PGA converted into glycolate. Tell me friends, what happened? Phosphoglycolate converted into glycolate. So what does it mean? It lost the phosphate group. That's all. So phosphoglycolate converted into the glycolate. Now the question asked in your 2025 question paper as one of the statement, how this glycolate entering into the peroxisome? This is the question. Now tell me Wagdevi, peroxisome having how many membranes? Is it a single membrane or double membrane? Peroxy, nucleus, mitochondria and chloroplast only having double membranes. Most of the organelles are what? Single membrane. Now tell me, peroxisome, tell me, glycolate can cross the plasma membrane? Glycolate can cross the plasma membrane or not? How come you forgot everything? We discussed it, right? Plasma. Through the plasma membrane, none of the positive charge and none of the negative charge cannot enter. Correct na? Plasma membrane is made up of what? Lipids. Lipid never allow any positive and negative. What about the glycolate? It's a negative charge. Hence it cannot cross the plasma membrane. Then definitely it requires a carrier. Correct na? That's the exact statement. Glycolate uses a specific carrier located on the peroxisome. Then glycolate, where it is entering? Into the peroxisome. The moment it enters into the peroxisome, glycolate converted into glyoxylate. Glycolate converted into what? Glyoxylate. Now glyoxylate converted into glycine. Right? Now this glycine will enter from where? From the peroxisome into the mitochondria. Glycine also will enter via specific transporter only because the membrane is not at all permeable. Correct na? Then glycine finally entered where? Mitochondria. Now this glycine can be converted into what? Serine. Serine is also an amino acid. Glycine is also an amino acid. Now what happened? Understand the picture. Whenever glycine converting into the serine, very important statement. Now all of you please look into this. It is what reaction? Oxidation or reduction? Reduction. NAD plus reduced to NADH. We generally gain energy. We got the energy here. Remember that step. When glycine is converted into serine. Now in the process of conversion of the glycine into serine, there is a release of ammonia. There is a release of what? Ammonia. What is the fate of this ammonia? We read it. This ammonia only enter into the nitrogen cycle, converted into amino acid again. But remember that point. Now the serine enters into where? Peroxisome. Now serine converted into what? Hydroxypyruvate. What is that? Hydroxypyruvate. Now hydroxypyruvate converted into glycerate. Now observe the picture. Now what is this reaction? Oxidation. We gain energy, we lost energy. Both equations got balanced here. Now glycerate again entered into where? Chloroplast. Now glycerate converted into 3-phosphoglycerate. How can we convert the glycerate into 3-phosphoglycerate? Just by addition of phosphate. The phosphate can be donated by the AT. So now I'll summarize everything, all of you focus. Whenever Rubisco fix the oxygen instead of the carbon dioxide, instead of getting 2 molecules of 3-PCA, we get only 1 molecule of 3-PCA and 1 molecule of 2-PCA. Now this 1 molecule of 2-PCA has to move into all the organelles. Again, they have to be converted into 3-PCA. Then only the Calvin cycle will be continued. So for that reason, this 2-PCA converted into initially glycolate upon losing phosphate group. Then the glycolate through the specific carrier enter into the peroxisome. Then glycolate converted into glyoxylate. Glycolate converted into glyoxylate upon combining with hydrogen peroxide. Glycolate combines with H2O2 converted into glyoxylate. Now glyoxylate converted into glycine. Glycine enters into the mitochondria. Glycine converted into serine. It is a reduction reaction. There is a production of NADH and there is a releasing of ammonia. This ammonia fate we will understand in the nitrogen metabolism. Now serine again enter into the peroxisome, converted into hydroxypyruvate. It entered into the, it converted into glycerate. Again it is what reaction? Oxidate. We gain energy, we lost energy. Then glycerate entered into the chloroplast. Upon spending energy, it is converted into 3-PC. So you have to remember every single reaction here. There is a great possibility we can expect the statements. Now tell me, which of the following cell organelles of the C2 cycle involved in the production of optically inactive amino acid? First understand my question. Which of the following cell organelles of the C2 cycle is involved in the production of optically inactive amino acid? What is optically inactive amino acid? Glycine. Why? Why it is optically inactive? Because it cannot rotate the plane polarized light. In other words, it does not have chiral carbon. Correct now? If you observe the structure of this particular glycine, it contains carboxyl group, amine group and one hydrogen. R group also contain hydrogen. When the carbon containing 4 different groups, then only it is chiral. Then only it is optically active. See here the question is about not optical activity, question is about organelle. Correct now? Which is the first intermediate that entered into the peroxisomes? Which is the first intermediate enters into the peroxisome? What is that? Glycolate. In which of the following cell organelles of the C2 cycle transamination reaction occurs? Slowly. Which is mitochondria, because glycine is converting into serine. We discussed that one amino acid can be converted into another amino acid by transferring of amino group. Okay, so the question they may link to the biochemistry and they will ask. So finally what we produced? 2 molecules of 3-PC. Correct now? So now here comes the important point. This C2 cycle only happens in the C3 plants. Okay. Now, why this C2 cycle never happens in the C4 plants and CAM pathway? What is the reason? Because C4 plants and CAM plants are having lot of adaptations, lot of escaping mechanisms to prevent the mechanism of photorespiration, right? Now, the most important pathway in the exam, for the exam point of view, C3 and C4. 90% they'll combine or compare and they will raise the questions. Okay. Even in the 2024 papers, question was specifically asked from the CAM pathway. Okay. Now, look into this. C4 pathway. We called it as the C3 pathway because the stable or intermediate product produced is 3 carbon. We are calling it as a C4 pathway. The stable or intermediate product is 4 carbon containing compound. We read that 97.5% of the plants on this earth belongs to C3 pathway only. Only those plants which are growing in the hot and dry climate, or the plants of angiosperms, or most of the monocots, they follow C4 pathway. The moment you heard the name of a C4 pathway, the common statement we all has to remember, C4 plants or C4 leaves follows Kranz anatomy. This is the first statement we have to remember. What is the most principal enzyme in the C3 pathway? Rubisco. When it comes to the C4 plants contain Rubisco or not? Cam plants contain Rubisco or not? Remember one common statement, every plant on this earth contain Rubisco, okay? But in some plants it is most important principal enzyme. In some plants it is present but it is not principal enzyme, right? We'll see how. In case of a C4 plants, the most important enzyme is PEP carboxylase, phosphoenol pyruvate carboxylase. Along with that, it also contains Rubisco. Rubisco is present. Without Rubisco, it is impossible, plant won't survive, right? So, but this cannot be the principal enzyme. Now, since in case of a C4 pathway, we are fixing the carbon into 4 carbon containing compound. That 4 carbon containing compound is what? Oxaloacetic acid. We'll remember that. So, these 4 are the standard examples of the C4 plants: maize, sorghum, millet and sugarcane. There was a question asked in the exam, what I told you just now, all the monocots generally follows C4 pathway. Dicots never follow the C4 pathway. In simple words, angiosperms only follows the C4 pathway. There was a question asked, there is one dicot plant which is an exceptional plant. Even though it is a dicot, it follows C4 pathway. The name of the plant is Amaranthus. Okay. Amaranthus is the name of the plant. Even though it belongs to the dicot category, it can follow the C4 pathway. Now tell me, what's the ultimate goal of the C4 pathway? Ultimate goal of any plant is fixing the carbon into glucose. And what is another challenge? Skipping or escaping this C2 cycle or the process of photorespiration. Correct now? How does it happen? How C4 plants fix the carbon into sugar very effectively, and how they are going to escape this C2 cycle? This is possible just because of the presence of Kranz anatomy. All of you please observe the picture so correctly. Kranz anatomy means there is an involvement of 2 cells, mesophyll and bundle. What are the speciality of mesophyll? Mesophyll cells are those type of cells where abundant amount of chloroplasts are present, which means all the chlorophyll molecules are present in the mesophyll cells only. Now look at the picture very correctly. If you observe the structure, this is the leaf and we have taken the cross section. It is an upper epidermis, lower epidermis. Lower epidermis contain a guard cell. In between there is a presence of stomata. Then what we are having? We are having mesophyll cell and we are having bundle sheath cell. What's the significant difference between mesophyll and bundle sheath? If you observe the picture of the mesophyll very clearly, what you can observe? They are always present closer to the leaf surfaces. Correct na? It is present near the upper epidermis and lower epidermis. So, they are present close to the surfaces. And what's the interesting point? There is a great intercellular spaces are present between one mesophyll cell to the another mesophyll cell. But if you observe the bundle sheath, you can see here, these are all bundle sheath. They are located very deeper of the cell. And is there any intracellular space? No. That is an important statement to be remembered. Bundle sheath cell generally surrounds xylem and phloem. These are xylem and phloem. They do not have intracellular spaces tightly attached with each other. Mesophyll cells are loosely arranged because they have a lot of intracellular spaces. But if the C4 pathway wants to work effectively, we require cooperative function of both mesophyll also as well as bundle sheath. Now, which means definitely mesophyll and bundle sheath cell has to connect with each other. How they connect with each other? Via plasmodesmate. What is plasmodesmate? Cytoplasmic bridge. Correct now? See, this is the cytoplasm of mesophyll cell. This is the cytoplasm of bundle sheath cell. Both are connected with each other via plasmodesmate. Without connection, it is impossible. The C4 pathway never takes place. Now, I'll explain slowly. All of you focus very correctly. What is our goal? Fixing the carbon into glucose and escaping C2 cycle. Right. Now interesting point here, there are two enzymes are involved, PEP carboxylase and Rubisco. Now look into this line, mesophyll cells which are closer to the leaf surface, they do not have Rubisco. Then instead of Rubisco they are having PEP carboxylase. What's the advantage of the PEP carboxylase? The advantage of the Rubisco is it can fix carbon dioxide also, but it's not necessary to fix the oxygen. This is one demerit of this enzyme. But what about the PEP carboxylase? It can fix only carbon, it cannot fix oxygen. Right, that is the advantage here. Now observe the picture, what exactly happening here. This is the atmospheric carbon, this is mesophyll, this is bundle sheath, and this is plasmode aspirator. Who accepted the carbon in the C3 pathway? Who is the carbon acceptor? RUBP. Correct now? How many carbon? 5 carbon. So someone definitely has to accept this carbon in the C4 pathway also. So who is the carbon acceptor in the C4 pathway? Phosphoenol pyruvate. Where we read this in the glycolysis, 9th step. Phosphoenol pyruvate is how many carbon? 3 carbon. Accepting 1 carbon from atmosphere. Then it becomes 4 carbon. What is the name of that 4 carbon? Oxaloacetic acid, which is the 4 carbon. Unfortunately, the oxaloacetic acid do not have any binding site to enter into the bundle sheath cell. That's the reason oxaloacetic acid converted into, we'll see that, either malic acid or aspartic acid. Both malic acid and aspartic acids are having the binding sites to enter into the bundle sheath cell. So initially malic acid enter into the bundle sheath cell. Malic acid is how many carbon? Oxaloacetic acid is how many carbon? 4. It is just converted into another form of acid, malic acid, that is also a 4 carbon. Now where the malic acid enter? Bundle sheath cell. Correct na? So malic acid is how many carbon? 4 carbon. Now in the bundle sheath cell, what is present? Rubisco is there in the bundle sheath. Correct na? Now this Rubisco break down this malic acid into 3 carbon plus 1 carbon. Correct na? By a mechanism called as decarboxylation. Then this 1 carbon taken by the Rubisco and continues the C3 cycle. How many carbons are left out? 3 carbons are left out, called as pyruvate. Now pyruvate coming back to where? Mesophyll cell. Upon spending ATP, it is converted into what? Phosphoenol pyruvate. Okay, I'll explain one more time. We'll see it. It's a very bigger one. Okay. First, I'll give you the overview, then we'll discuss step by step. So first, atmospheric carbon dioxide accepted by phosphoenol pyruvate, 3 plus 1 carbon converted into 4 carbon, giving rise to oxaloacetic acid. Since it do not have an active site, is converted into malic acid or aspartic acid. Now malic acid, which is a 4 carbon entering into the bundle sheath cell, this undergo the mechanism of decarbonation, breakdown into 3 carbon plus 1 carbon. Now that 1 carbon taken by Rubisco and continue the C3 cycle. That remaining 3 carbon called as pyruvate transported back to the mesophyll cell and converted into phosphoenol pyruvate. Now my question is, anyway Rubisco where it is located? Then why Rubisco did not fix the oxygen, why it only fixed carbon dioxide? Because we know that Rubisco having the ability to fix the carbon also, oxygen also. Then why it fixed only carbon? Because where the bundle sheaths are located deeper. Correct na? Whichever the cells located the deeper, they are having poor access to oxygen. Oxygen only cannot enter inside. Hence Rubisco successfully fixed only carbon into glucose. Correct na? Now we will understand step by step elaboratively. Okay. There are 4 steps are involved here. Number 1 is called breakdown, splitting, phosphorylation. Very simple, all of you understand. First, carboxylation means what? Tell me. Addition of carbon. So we are adding 3 carbon with 1 more carbon. Correct na? 3 carbon phosphoenol pyruvate, 1 carbon dioxide from atmosphere. What do we got now? Oxaloacetic acid. What's the enzyme catalyzed this reaction? PEP carboxylase. Since it is a carboxylase enzyme involved in the carboxylation reaction. Right. Next, breakdown. Why we have to break down? Since oxaloacetic acid cannot enter into the bundle sheath, so I am converting oxaloacetic acid into either aspartic acid or malic acid. Right. Oxaloacetic acid converted into aspartic acid by an enzyme called transaminase enzyme. You have to remember. Similarly, oxaloacetic acid converted into the malic acid by malate dehydrogenase enzyme. Okay. Now my simple question, tell me, what is the location for carboxylation and breakdown? Which cells it happened? Carboxylation happened in mesophyll. Breakdown happened in mesophyll only. Correct na? Then malic acid entered into the bundle sheath cell. Now what is the reaction? Splitting. Correct na? What is the reaction? Splitting. Now malate is how many carbon? 4 carbon. Breakdown into 3 carbon plus 1 carbon. So this is what reaction? Decarbox. So enzyme is decarboxylase. Correct na? Now this 1 carbon, who will take it? And continue which cycle? Calvin cycle. Now whenever the carbon dioxide taken by the Rubisco and continuing the Calvin cycle, definitely it produce 1 glucose. So how many ATP we required again? 18 ATP, 12 NADH and 6 carbon. This is same. There won't be any change. The only change occur here. Now what is happening here? This 1 carbon taken by the Rubisco and continue the cycle. How many carbons are left out? Which is called pyruvate. Now this pyruvate is located in which cell now? Mesophyll or bundle sheath? Bundle sheath. So it has to enter into the mesophyll cell. Now look into the point, no confusion. Pyruvate entered where? Mesophyll cell. Converted into phosphoenol pyruvate. Correct na? Converted into phosphoenol pyruvate. Now this is the fixed. Pyruvate, in order to convert it into the phosphoenol pyruvate, every time it requires 2 ATP. How many? Now all of you tell me, in order to produce 1 glucose, how many times cycle has to turn? 6 times. 1 time we uses 2 ATP. So 6 times 12. So in the C4 cycle, 12 plus 18, 30 ATP, 12 NADH and 6 carbon dioxide. But in case of C3 cycle only 18, 12 and 6. Now my question is, in order to convert the pyruvate into the phosphoenol pyruvate, generally we require only 1 ATP. You remember the 10th step of glycolysis? Phosphoenol pyruvate converted into pyruvate. There is a loss of 1 phosphate only. That converted into 1 ATP. Then why I have written 2 ATPs here? The answer is the enzyme is dikinase. What is that? This is also a question. Okay. These all won't be present in the books. Okay. We have to understand that correctly. What is that? Dikinase. So which means every time it uses 2 phosphates. So how many times pyruvate converted into phosphoenol pyruvate? 6 times. Because cycle has to turn 6 times. So we definitely uses 12 ATP. Then in case of C4 cycle we require 30 ATP, 12 NADH and 6 carbon dioxide in order to produce 1 glucose molecule. Is there any C2 cycle occurred here? Is there any photorespiration? No. That's why we can say that C4 plants are more efficient in fixing the carbon compared to the C3. Even at a higher temperature. That is the question here. That is important. If they ask you generalized conditions, no higher temperature, definitely C3. Only in case of higher temperature or high light intensity, C4 are more efficient in terms of carbon fixation. How can we identify which plants are more efficient in carbon fixation? There is a simple question, by using isotopes. What is that? By using isotopes, that we'll see in the upcoming slide. By using isotopes, we can understand which plants are more efficient in the fixing of carbon. Right? Clear everyone so far? Okay. All right. Okay. Clear everyone online? Remaining will continue tomorrow. Okay, take 10 minutes break. We'll continue the biochemistry class 1. Okay. Okay, take 10 minutes break everyone and please join. I'll share the link. Okay. All right. Thank you all. Thank you. Okay, take some break.